Ladies and gentlemen, please welcome Genome BC's Vice President of Communications and Societal Engagement, Sally Greenwood.
Good afternoon, everyone, and thank you for joining us for today's Genome BC's annual Don Rix Distinguished Keynote address. My name is Sally Greenwood. As you now know, I am the Vice President of Communications and Societal Engagement for Genome British Columbia. Let me begin by acknowledging that we are gathered on the traditional, ancestral, and unceded territory of the Coast Salish people who have been the custodians of this land for thousands of years. I would also like to welcome students from Gladstone, Heritage Woods, and Chilliwack Secondary Schools today. These students are the researchers, teachers, artists, business leaders, physicians, and entrepreneurs of tomorrow. Like the generations before you, it is your aspirations that will provide the new innovations and opportunities that will benefit future generations, much like Don Rix, the person for whom this event is named. Let me tell you about him. Don Rix was an innovator and a philanthropist with a broad palette of interests: medicine, science, education, and the arts. He was a recipient of the Order of Canada, the Order of British Columbia, and one of the co-founders of Genome British Columbia. Don believed that technology and innovation were the fundamentals upon which to build the future of British Columbia. In a speech to the Vancouver Board of Trade, he explained that technological innovation transcends all boundaries. He said, quote, "Not only does it create new industry sectors and new jobs, which in turn boost economic output, it also provides traditional industries with advances that allow them to be more productive and competitive. More importantly, technology improves every aspect of life by coming up with new ways to address important societal issues." Don was a visionary. He was a pioneer. Throughout his life, he looked for gaps and created opportunities to fill them. One of the things we know about Don is that almost every new startup or life sciences industry or company in Vancouver today was started with a small investment from him personally. He was opening his wallet all the time because he believed so strongly in what he was doing. As a physician, he saw the need for better diagnostics and helped to co-found a system of clinical labs across Canada, better known today as Life Labs. Don had a keen interest in biotechnology, and as the chair of our board through our formative years, his influence on Genome BC is still felt today. We should never lose sight of his vision or his spirit of collaboration. This has guided our trajectory over the last 20 years. When we lost Don in 2009, we decided to recognize his legacy by establishing the Don Rix Distinguished Keynote Address to continue to inspire our community. I believe that Don would be very happy and approve of the keynote speakers we have had to date, and today is no exception. Let me thank you for joining us today as we continue to learn and explore the transformative science of genomics. Before we get started, a few housekeeping things. You will notice on your screen that we have different ways for you to track and interact, so please make note of those, and we'll keep them up on one of the screens throughout the presentation. After the keynote address, Dr. Pascal Spothelfer, our President and CEO, will moderate a 30-minute Q&A, so hold your questions until the end. And one way that you can ask questions if you don't want to come up and take a microphone is, again, to use Slido, which is here. Over the details of how to connect are over here. You can also participate in the social conversation on Twitter, #DRDK2019, and again, details are behind me. Following our keynote address, you're welcome to join us for a reception outside the doors in the mezzanine. Please wait until the Q&A has concluded before rushing out the doors. At this time, I would like to ask Pascal to come forward and to introduce this year's keynote. Thank you very much.
Thank you, Sally. Good afternoon. Our vision statement says, genomics positively impacts life every day. This may be a future state. To get there, we need contributions from so many people, from here, at home, and around the world, who move this technology forward. These efforts go beyond biological research and include investigating questions at the intersections of genomics and society. And this is why we organize events like this, where we engage in a dialogue that increases our collective understanding of this transformational science. And this event serves exactly this purpose. Over the years, we have profiled many genomics pioneers whose work are revolutionizing. Today's talk is no exception. Dr. Claire Fraser is a pioneer and global leader in genomic medicine. From 1998 to 2007, Claire was president and director of the Institute for Genomic Research in Rockville, Maryland, and led the teams that sequenced the genome of several microbial organisms, including important human and animal pathogens. She helped launch a new field of microbial genomics and revolutionized the way microbiology has been studied. Her work with the FBI on the Amerithrax investigation between 2001 and 2008 led to the identification of 4 genetic mutations of the anthrax spores that allowed the FBI to trace the material back to its original source. She is one of the world's experts in microbial forensics and the growing concern about dual use, research that can provide knowledge and technologies that could be misused. Claire has authored more than 300 publications, edited 3 books, and served on the editorial boards of 9 scientific journals. For 10 years, she was the most highly cited investigator in the field of microbiology. Claire is currently the Dean's Endowed Professor at the Department of Medicine and Director of the Institute for Genome Sciences at the University of Maryland School of Medicine in Baltimore. And earlier this year, she was chosen as president-elect of the American Association for the Advancement of Science, the world's largest multidisciplinary scientific society and the leading publisher of cutting-edge research through its Science family of journals. Today, Claire is going to help demystify the role of the human gut microbiome in health and disease. And so, it is with great honor that I welcome Dr. Claire Fraser to speak at our 10th Don Rix Distinguished Keynote address. Please join me in welcoming Claire.
Thank you. That was lovely. Thank you. Pascal, thank you for that very gracious and generous introduction. I can't tell you how delighted I am to be the guest of Genome BC the last couple of days. I didn't have the privilege of knowing Don Rix, but from what I've heard and what I've read, he was truly a visionary and someone who worked to make a difference every single day. And I particularly like the Genome BC slogan, if you will, making a difference every day. I wish we could somehow I wish I could take that back to Maryland and use it, but perhaps we'll modify that, because I absolutely believe that the field of genomics has been transformative and the best is yet to come. So I want to thank all of you for coming today. I particularly want to thank all the students. And I hope that in the time that we have together, I can share some of what I have learned together with my colleagues over the last decade in our study of the human microbiome, particularly the human gut microbiome. And I hope at the end you will go away a little bit more convinced than you might have been when you arrived, that this truly is an exciting new area of research and one that I think ultimately will transform the practice of medicine. So let me begin by just asking you to think about an ecosystem. And I'm sure there are lots of different things that come to mind. For some of you, you might have thought of a rainforest. For others, you might be thinking, an ecosystem, that's a coral reef. I wish we could say that these are thriving ecosystems, less so today than they were in the past. Another example that's a favorite of mine is Mr. White's terrarium that he planted and watered and sealed almost 30 years ago. And here it is today, doing extraordinarily well. Another example of an ecosystem. But what we're going to spend our time thinking about today is what I think is one of the most important ecosystems of all, and that's the human microbiome, the collection of organisms that are with us from the day we're born until the day we die. And so just so we're on the same page, and so you don't feel too intimidated right at the start of this talk by thinking, oh my gosh, ecosystems, I think I have a general idea, but I don't really know what it is. For the purposes of what we're going to discuss today, we can consider an ecosystem in a very simplified way. An ecosystem is a group of living things plus all the non-living things that they need. And one of the features of an ecosystem is all the components are linked together. And probably the most important of all is that ecosystems are dynamic. They are continuously changing in response to multiple factors. So we have inputs to ecosystems. Within the ecosystems, we have the organisms and the processes that they carry out. And we have the outputs. And very often within ecosystems, you have multiple feedback loops. So for the purposes of today, you don't need to think of an ecosystem as being any more complicated than what I've just diagrammed in the slide. And as I said, the ecosystem that we're going to focus our attention on today is the human microbiome, the community of organisms, bacteria, viruses, archaea, protists that live in association with humans. And the microbiome includes not only these organisms, but all of their genetic material. So let me ask a question before I go on to the next slide. How many of you, and just by a show of hands, how many of you would guess that this concept of the human microbiome was discovered in the past 20 or 25 years? Show of hands. Okay, a few. The last 50 years. The last 100 years. And longer than 100 years. So there's a fairly even distribution there. And as is so often the case in science, it is hard to come up with an original idea. And I think that we can trace the idea of the human microbiome, if we define it as the collection of organisms that live in association with humans, back to von Leeuwenhoek, who has been called the father of microbiology and lived from 1632 to 1723. You may remember that he is probably best known for his invention of the microscope, which was truly groundbreaking with regard to the field of microbiology, because prior to this, all of the microbes present on Earth were not able to be seen by the naked eye. And von Leeuwenhoek was so excited by what he saw in the first few samples that he continued to look at samples. He continued to sample environments. And he didn't find an environment where he didn't see evidence of microbial life. And what I think is so interesting and so amazing, and it really sort of reflects the fact how if you wait long enough, science often comes full circle. One of the samples that got him most excited was the material that he scraped off of the surface of one of his teeth. And if you think back in the late 1600s, oral hygiene was probably not as advanced as it is today. So my guess is there was a lot of stuff that came off of the surface of his tooth. And what you see here, what you see here are some of his drawings of the different kind of microbes that he observed. And he was so excited by what he saw that he wrote a letter to his colleagues at the Royal Society of London describing this diversity of microbial life. He didn't know what to call these organisms. The term bacteria had not been coined yet, and so what he called them animalcules, and he wrote that the animalcules were present in such enormous numbers that the water seemed to be alive. So I think we really need to credit von Leeuwenhoek with the first discovery of the human microbiome. And I think it's interesting because the microbiome that resides in the oral cavity is one of the human environments that has received a lot of scrutiny using the current tools. We're going to go through just a little bit more history. Trust me, I'm not going to take you through 400 years. But I would say that the next big breakthrough in the field of microbiology and also the study of the microbiome came from the work of Louis Pasteur, who lived about 150 years later than von Leeuwenhoek and has been called the father of microbial culture medium. He did a lot of incredible things. He promoted the idea of vaccination, the process of microbial fermentation, the process of pasteurization to preserve foods. But one of the other things that he developed was liquid culture media in which to grow microorganisms. So now the field of microbiology could go from just looking at organisms under a microscope to actually growing them in the laboratory. And I would say that these two discoveries really positioned us for a considerable period of time. And not too many additional breakthroughs happened for the next 100 years. And based on this foundational work of people like von Leeuwenhoek and Pasteur, it was really deemed that pathogens, disease-causing microbes, were the most important microbes and the ones most relevant to human health. And because we didn't have the tools to really study these microbes in much detail, the conclusion was that the population of microbes in and on the human body was really pretty mysterious. And I think some erroneous assumptions were made. The normal flora, as they were called for a long time, were considered to be mostly inert. It was thought that they were mostly stable. And it was also thought that they weren't relevant to disease. I would say the next big breakthrough came in the middle of the 20th century through the work of Rene Dubose, who was a French-born microbiologist who spent much of his career at Rockefeller University in New York. He actually received a Lasker Award for his work on soil microbes and antibiotics. But most relevant to the study of the microbiome was the groundbreaking work that he did with germ-free mice. These are mice that are raised in sterile conditions, and so as a result are not colonized by any microbes. And it was through his work with germ-free mice that I think we really came to understand how important our microbial partners are in health. One other interesting, he was truly a Renaissance man. He won a Pulitzer Prize for nonfiction for a book entitled So Human an Animal, and it's thought that he was the person who coined the phrase, think globally, act locally. But his insights that came through his studies on germ-free mice, and this is an excerpt here from one of his other books called The Mirage of Health, says these microorganisms should not be regarded merely as contaminants, meaning contaminants of the human body. Rather, they become so intimately associated with the various digestive organs that they form with them a well-defined ecosystem of which each component is influenced by the others and by environmental conditions. And those words are as true today as they were back in 1959. And if we fast forward then to where we are today, I think so much of our understanding of the human microbiome has come from work over the past 15 years or so, work that my colleagues and I and others have done using genomics-based approaches to study the microbiome. What these approaches have allowed us to do is bypass the need of growing any of these organisms in culture. We can go into whatever environment we are interested in, sample the microbes, extract the DNA, the RNA, and the proteins, and use these large-scale approaches to ask questions like, who's there? What is it that these organisms are doing? How do they respond to various external factors? How are they interacting with us as a host? And what are the chemical outcomes of their activity? So that's the extent of my little history, if you will, of the microbiome and how we got to where we were today. But like so much of science, this really has represented a series of breakthroughs. And those of us who are practicing in the microbiome field today, building upon the work of many others who have come before us. So let's summarize what we think we know about the microbiome before we get into some details. The estimates are that there may be as many as 100 trillion bacteria, fungi, protozoa, and viruses that live on and inside the human body. It's a little bit hard to grasp the size of that number, but it is enormous. There have been some debates about what this number really is. Some at one point thought there were maybe as many as 10 times the number of microbial cells on us and in us as humans as there are human cells. That number has been revised downward to maybe 3- to 5-fold. Obviously, this is based on all sorts of assumptions and estimates. Nobody's gone about trying to count all of this. But I think what is indeed correct and what is really important is that if you look at the number of microbial genes, and we can make those estimates fairly precisely, and compare those to the number of genes we now know exist in the human genome, we think that there may be as many as 150 times the number of microbial genes as there are human genes. So that means that a considerable amount of the biology that we attribute to being human biology really represents the biology that is contributed by our microbial partners. And we know that they carry out a number of essential processes, and this is by no means a complete list. They help us extract energy from our food, they produce essential vitamins, they regulate our immune system, they regulate various aspects of our metabolism, and they also protect us against disease-causing microbes. And as I said earlier, personal microbial communities shift throughout a person's lifetime and can be influenced by a number of different factors, which we will cover later on in the talk. So how did this all begin? When did we first set up this association with our microbial partners? Well, we know, my apologies again, we know that life on Earth can be divided into 3 major domains: the bacteria, the archaea, and the eukarya. The eukarya are, for the most part, multicellular organisms. There are some simple single-celled eukaryotes, but we as humans sit up here at the very tip of this branch of the phylogenetic tree of life. But bacteria preceded humans on Earth by billions and billions of years. So when multicellular life evolved, the bacteria were already here waiting to set up associations and to colonize all of the multicellular organisms. So these ecosystems, the development of these ecosystems really was facilitated by the fact that bacteria preceded us. And bacteria are incredibly diverse and resilient and can colonize almost every environmental niche that you can think of. So once this association was set up, and this picture here is really meant to depict the coevolution of us as humans and our microbial partners, we then needed a way to perpetuate this. And what we have come to really understand is that we have vertical descent of our microbiota through the process of birth. And we will explore this process of colonization in the next few slides. We understand that infants acquire their initial inoculum of microbes that go on to develop into the adult microbiome from our mothers. And there are two different ways that this can happen. Typically, this happens through normal delivery, in which case the infants pick up an initial inoculum of microbes that looks very much like the community of microbes that exists in the vaginal canal. But in some cases, cesarean sections are necessary. And that means that the babies don't go through the birth canal. And it also means that they are inoculated with a different set of microbes. They tend to look more like the microbes that are found on the mother's skin. And a number of studies have really followed what happens after birth depending upon the mode of delivery, whether it's a vaginal delivery or a C-section. And these are data from a very recent paper that was published last month looking at close to 800 infants, half of whom were delivered with vaginal delivery and half through C-section. And it looked at the composition of the infant gut microbiome at 4, 7, 21 days following birth, and then towards the end of infancy at close to a year. And you can see what these represent here are stacked bar charts. Each color represents a different genus of bacterial species. We don't need to get into the details of who or what they are, but what you can see is that babies born vaginally display a fairly consistent microbiome composition over the first year of life. There are some shifts, but overall these microbiome data are fairly stable. Contrast this with what you see with babies born by C-section, where there is a much more dramatic change in the overall composition of the microbiome. And by about a year of age, it looks as if you compare these data to this last bar chart here, that the mode of delivery really doesn't matter because the C-section babies tend to begin to resemble the vaginally delivered babies. However, there have been a number of studies looking at whether or not there is a consequence to how babies are delivered, and there are some intriguing links that have been reported. And this will be the first of a number of links that I'm going to talk about. And I want to caution that at this point in time, these are associations. These data do not in any way speak to cause and effect. And I think this is because the field of microbiome research is really in its infancy, pardon the pun if we're talking about infants here. And so we are really in an observation stage. We're collecting data. And these observations are allowing investigators in the field now to generate hypotheses that they are going on to test. But some of these links between C-section births and later clinical outcomes are really pretty interesting. A 2015 paper that looked retrospectively at almost 2 million Danish children from infancy to 15 years of age reported that there was an increase in the incidence of asthma, connective tissue disorders, arthritis, inflammatory bowel disease, leukemia, and other immune deficiencies in infants that were delivered by C-section. 2012 study looking at a little over 1,200 children in the U.S. At 3 years of age suggested a link between C-section delivery and obesity. And a 2003 paper, so these studies go back a ways, looked at 2,800 Norwegian children and reported an increase in various food allergies in infants born by C-section. Now, in no way am I up here suggesting that C-sections don't play a critically important role in modern healthcare. In some situations, they are absolutely indicated. But certainly in the United States, and I don't know if it's as true in Canada, it is thought that there are many C-section deliveries done more as a matter of convenience than medical necessity. So I think some of the results from these microbiome studies are giving us pause to think that maybe it's worth reconsidering these decisions if indeed we can go on with follow-up studies that go beyond just associations to demonstrate, in fact, that these links are real. There has been a suggestion that it may be possible to partially restore the microbiota of C-section-born infants through vaginal microbial transfer, taking a sterile gauze, saturating that with fluids from the vagina, and rubbing down C-section babies immediately after birth. This suggestion, this report, was immediately met with a great deal of enthusiasm, but I would say that enthusiasm has been tempered because there are also some risks associated with this in transferring pathogenic microorganisms, and that would be an unintended consequence. But I guess I just want to say before we move away from this topic that this is an area of considerable interest in terms of microbiome research that's underway currently. The next thing that I want to talk about, and I think is another very important determinant of microbiome development, is the early diet. Specifically, breastfeeding versus formula feeding in the first several months of life. In similar studies to the one that I just showed you about C-section deliveries versus vaginal deliveries, there are many studies that have demonstrated that the organisms that develop over the first few months of life in the infant gut microbiome differ, depending on whether an infant is breastfed or bottle-fed, formula-fed. And I think that, to me, one of the most fascinating aspects of all of this is the fact that if you look at some of the chemical structures that are found in human milk, you see that there are classes of these complex human milk oligosaccharides. And what I think is so fascinating is that human infants do not have any of the enzymes to break down these human milk oligosaccharides, which can represent a significant part of human breast milk. But instead, what we see is that some of the key species in the infant gut microbiome, particularly a number of species of bifidobacterium, encode all of the necessary enzymes for breaking down these milk oligosaccharides and extracting energy. So I think this is really a beautiful example of coevolution, of how the constituents of human milk appear to have co-evolved in a way where they serve as the ideal source of nutrition for the microbes that are in the human infant GI tract. And what we know is that despite their best efforts, companies that manufacture infant formula have had a difficult, if not impossible, time in replicating this enormous array of human milk oligosaccharides. So let us go on then. And you might ask, well, does all of this matter? If delivery mode affects the type of organisms in the infant gut in the early months, and if the diet affects the early microbiome composition, does it really matter if these communities eventually converge? And again, this is just speculation. This has not been demonstrated. But over that same time course during which the human infant microbiome is maturing, so too are a number of important organ systems. The human brain is finishing its development, and the human immune system is also maturing over that same time course. And what we have come to understand is that probably one of the most important early interactions between the infant gut microbiome and the immune system takes place in the GI tract. That's where the largest part of the human immune system resides. Prenatally, all the foundations of the immune system are present. What this diagram is meant to illustrate is a schematic of the intestinal tract. But as soon as the GI tract is colonized following birth, we see a rapid expansion and maturation of the human immune system. This doesn't happen in germ-free mice. And what we have come to believe is that one of the most important outcomes of this interaction between the infant gut microbiome and the gut immune system is the ability of our immune system to recognize our microbial partners as self. And therefore, they don't react to them the same way they would react to pathogenic microbes that they might encounter in other circumstances. And again, another set of correlations only, but interesting ones. Through lots of public health efforts over the past 60 years or so, we have seen a dramatic decrease in the rates of infectious disease, but we have also at the same time seen a dramatic increase in the rates of various disorders that are thought to have an autoimmune component. And so it raises the question, have we, through lifestyle choices, whether necessary or mandated because of medical situations, ended up becoming too clean, if you will, too sterile? And I think a great read from Vancouver's own Dr. Brett Finley is this book here, Let Them Eat Dirt, which talks about some of the potential pitfalls of rearing children in an over-sanitized world. So there is much to do here, but these are intriguing correlations. And finally, by the time a baby gets to be a toddler, about 2 or 3 years of age, the gut microbiome has taken on the characteristics that look very much like the adult microbiome and will carry through for the rest of this infant's life. So then you might ask, and one of the major areas of focus of the early microbiome efforts was to define a healthy human gut microbiome. And you might think, well, this is simple to do. We'll just look at the microbiome and characterize it in healthy individuals. But Brett Finley and I were part of this group that convened back in 2012 as the Human Microbiome Project was nearing the end of phase 1 to ask the question, are we close to being able to define a healthy human microbiome? And the reason for doing that, I think, is important and probably very obvious. And as we concluded in this paper that the group published, elucidation of the properties of a healthy microbiota would provide a target for dietary interventions and microbial modifications aimed at sustaining health and improve the health status of people exhibiting disrupted microbiota and associated diseases. What you might glean from this comment, and what I didn't pull out of this paper, was a statement that concluded that yes, we indeed know what a healthy microbiota looks like, because that has turned out to be a bit of a challenge. One of the things that came from the early studies of the human microbiome was the appreciation that across different body sites, if you look at either of these two individuals here, and these pie charts represent relative abundance of different types of bacteria. So across human body sites, the microbiota in various environments differ. That's not really so surprising. I think what was much more surprising to all of us was the fact that if you look across individuals, and again, you can see that with these pie charts, and just let's take a look here at the colon in individual A and the colon in individual B. These two communities, based on who's there, aren't at all similar. And there have now been tens of thousands of microbiomes that have been characterized from healthy individuals. And these are just some early data from my lab. And what this represents is a series of about 400 stacked bar graphs, again, looking relative abundance of different microorganisms that are commonly found in the human gut. And I think what you can immediately conclude is that there are no two individuals where the composition of the microbiome is the same. And if you look over time, it's not just looking across individuals, but if you look over time, in a given individual, we see that there is both short-term and long-term variability in the human microbiome. And it gets back, I think, to that fundamental principle of ecosystems. They are inherently dynamic. And we now have a much better understanding of what factors are at play in modulating these changes in microbiome composition. And this is a slide that just touches upon some of these, and I'm not going to go through all of them, but you can see that they are enormous. There are factors intrinsic to the microbiome, factors intrinsic to us as the host, and then all sorts of extrinsic factors related to the host and the environment in which we live. And if you look at all of this, again, this really gets at the fundamental idea of what an ecosystem is and how it operates. So what we have come to conclude, and this is one of my cautions, particularly when you read about new studies related to the microbiome in the press, I think we have decided that looking at who's there and looking at that across individuals or over time is probably not the right thing to be measuring. And so instead, I think where the field is now going is to begin to look at function. What is the function of these microbes? How are they working together to carry out the functions that are essential for human health? And these are pie charts that represent data from another recently published paper. On the left, you will see a pie chart that captures the species, the types of organisms that are shared across individuals. And you will see that half of them, 50% of them, are only found in 1% of all the samples looked at. If you take the next biggest part of this pie chart, almost 80% of the microbes are only detected in 25% of the subjects. But yet, if you look at the microbiome through a different lens, a functional lens, looking at biochemical pathways, at metabolic pathways, you see a picture that's almost completely flipped, where in this case then, over 50% of the pathways, nearly 75% in some cases, are found in the majority of samples. So I think what this means is that what it says is that we have many tools at our disposal to look at microbiome, but who's there is probably not the right thing to be looking at if we really want to understand health and disease. We should be looking at what these organisms are doing. And we know from basic ecology there is this concept of functional redundancy in ecosystems. You can have your number of species increase and ecosystem functions will increase, but in every ecosystem this eventually plateaus. Ecosystem functions are finite, but different combinations of organisms can carry out the same function, and that's what really matters. So I just want to spend the next few minutes that I have talking about a couple of external factors that I think are critically important. And these represent factors and situations that all of you can really do something about and think about it. And the first is the impact of diet on the microbiome. This quote was attributed to Hippocrates, but many have said they don't think he ever said this. But it's a great idea in terms of what we're talking about here. Let food be thy medicine, and medicine be thy food. And why this is so relevant is because there is abundant medical evidence that our diet greatly influences health. And for the longest time, we were trying to make inferences about why this is the case based just on what we know about human biology. But I think a lot of additional insights related to diet have come when we bring the microbiome into the equation. It's amazing to me how quickly a number of authors have jumped into this game to write about the best microbiome-based diet. I'm not going to point any fingers here. Some of these books are better than others. Some of these I don't think should have ever been published. But it really leaves you as consumers wondering, what in the world should I do? What advice should I believe? Well, maybe we can think about this from a very simple perspective in terms of the metabolic fate of a meal that you eat. And let's assume that this is a meal that contains all the major macronutrients: protein, fat, and carbohydrate. Sorry about that, I keep doing that. In the stomach, the process of digestion begins, and proteins begin to be dissolved, as well as fats. Most of the digestion of fats and proteins takes place in the small intestine, and when they are sufficiently digested, absorption takes place. But for complex carbohydrates, nothing happens until this material reaches the colon. And then this amazing collection of microorganisms in the colon goes to work to ferment these complex carbohydrates. And when I'm talking about complex carbohydrates, I'm really talking about what we can consider as fiber-rich foods. And one of the things that we know is that the microbes in the colon have the ability to break down these carbohydrates, extract the energy that they contain. But more important than anything, I think, is that one of the byproducts of this fermentation process is butyrate and other short-chain fatty acids, which are some of the most beneficial compounds that our gut microbiome produces. Butyrate has been shown to be a potent anti-inflammatory compound. If we take in too much fat and protein, we can overwhelm the ability of the system in the small intestine, and some of these undigested proteins and fats will make their way into the colon. And there are microbes in the colon that have the ability to work on proteins and fats. But unlike what happens with fiber, what is often produced are various toxic compounds. So be cautious about a meal that's high in protein and high in fat. You will pay the price in terms of adverse health consequences if these make it all the way to the colon. But the process is far more complicated than that. I'm not going to go into detail in this slide, but this is meant to be an overview of all of the incredible metabolic processes that take place. And here what we're looking at are butyrate-producing bacteria versus bacteria that break down dietary fat and phospholipids. And again, getting back to that notion of functional redundancy, we know that there are multiple carbohydrate metabolizers in the colon and multiple fat and protein digesters in the colon. So just because my microbiome may have a different taxonomic composition than yours, it doesn't mean that these same processes aren't being carried out. And to just focus on these two examples in a little bit more detail, I mentioned that the production of short-chain fatty acids from high-fiber foods leads to beneficial health consequences. Short-chain fatty acids have been shown to maintain the integrity of the epithelial cell layer in the gut, and they have a potent anti-inflammatory property. Contrast this to what happens if we eat a lot of red meat. Different sets of bacteria work on these compounds, and what is produced are compounds like trimethylamine that are further modified and have been shown to promote atherosclerosis and cancer. So you think about it, we learned years ago that we should cut back on our consumption of red meat because it promotes heart disease and cancer. And yet, really, the effects of that are all mediated through microbes in our GI tract. And even more complicated than this is that these metabolic effects extend beyond the GI tract. And I'm not going to go into this except to say that many of these end products, these metabolites, can get into the bloodstream and can have effects distally and affect multiple organ systems throughout the body. So I'm going to skip that there in the interest of time. And so you might ask, based on this, and I get asked this question all the time, what is the best diet to be eating? Well, I think unfortunately we know the answer in part. It's the diet that perhaps we don't want to eat. It's a diet high in fresh fruits and vegetables, low or no processed food, minimum amounts of red meat. But what we're also now finding is that because of the individual differences in the gut microbiome, it's probably going to be difficult to say that there is one best diet for everybody, because we are now beginning to understand that individual differences in all sorts of factors, including our microbiome, but our own host genetics, activity and exercise, other lifestyle factors, have an impact on our response to specific foods. And there are already companies that have come into existence that I think are actually doing some really interesting work, trying to take all these factors into account. And this one company here, Day 2, which is a company in Israel, was set up initially to try and help type 2 diabetic patients optimize their dietary intake. And what they found in a large-scale trial was that different individuals respond differently to foods. And there are some people where a piece of white bread will cause a spike in blood glucose, and in other people, blood glucose levels will stay relatively stable. They saw this with ice cream, they saw this with all sorts of things. So I don't think it's out of the question to think that we are perhaps moving towards a time when, with a better understanding of how the microbiome contributes to diet, that we will really be able to come up with individual recommendations for the most healthful diet for one individual. So I know I have gone way over time, and I'm just going to take a couple more minutes, if I can, and talk to you about antibiotics and the human microbiome. At least in the US, these are data from the CDC. It's thought that there are over 50 million unnecessary prescriptions for antibiotics each year. And this is a problem because antibiotics not only target the pathogens for which they were intended, but they also target the human microbiome. And I think about antibiotics in the human microbiome as almost being analogous to dropping a nuclear bomb on our microbiome. And in children in the United States, and these are old data from 2010, but they're still, the numbers haven't really changed that much today. The top 8 prescriptions in the US are shown here on this slide, but 5 of them are antibiotics. And this represents a total of over 41 million doses of antibiotics in kids per year. And not surprisingly, antibiotics have multiple effects on the gut microbiota. And maybe the most important one here is just the depletion of bacterial diversity. Studies have shown that following a typical course of antibiotics, the total number of bacteria can decrease by tenfold, and the diversity of organisms can go down even more. And add on top of that the concern about overuse of antibiotics and the development of antimicrobial resistance, and you really have a prescription for disaster. And what we have come to appreciate is that these effects of antibiotics can be cumulative over the course of a lifetime and cumulative over multiple generations. These are some interesting data from Marty Blaser, who is now at Rutgers, who has suggested that repeated administration or exposure to antibiotics through our food supply, for instance, over time leads to decreasing diversity in the microbiome. And if you remember that we inherit our microbiomes from our mother, this loss in diversity is passed on down through generations. Some interesting studies in mice, with the caveat that mice are not humans, have clearly shown that taking antibiotics early in life leads mice prone to obesity. And so it raises the question, does it not, that perhaps maybe the same thing is playing out in the human population. And again, we have studies that represent associations, but studies in fairly large cohorts of children that looked at the impact of early life antibiotic exposure with BMI and obesity later on in life. And what we found, what the scientific community has found, is that there are indeed these links between early life antibiotic exposure. Not something to be proud of, but these are data from the CDC looking at the percent of obese adults in 2010 and the rate of antibiotic prescriptions in 2010. And again, this is just a correlation, but I think the data are pretty striking, that the highest levels of obesity are present in regions of the country where antibiotic prescriptions are prescribed at increasing levels. So I know Pascal wants to get to questions, so I'm going to just, if you will permit me, just skip some of the last slides. What I'm not going to talk too much about, because I think the data are just so early on, are the potential links between changes or dysbiosis, as it's called, in the gut microbiota and various diseases. But I think it is, again, really quite compelling if you look at some of the diseases that are associated with aging, chronic diseases that have a component of inflammation, diseases of the nervous system, the cardiovascular system, the digestive system, cancer. And then if you ask, are there any data to suggest that there may be links between these chronic diseases and changes in the microbiome, particularly the gut microbiome? The answer is an unequivocal yes. And so what we don't know, and I will say that these results, and I've generated some of these myself in my own lab, but these results do not yet represent a smoking gun, but they raise all sorts of interesting questions. Perhaps the most important one is what came first, the disease or the change in the microbiome? And those are going to be tough questions to answer, but they can be done in human studies, and we can also make use of information from animal models of various diseases. But I will say that we know that the economic burden on health spending as a result of these chronic diseases is increasing at an unsustainable rate. I've called out two countries here, and I know you can't read them. Here's the US at the very top. We like to be on top, but not in a situation like this with regard to global health spending. The other arrow represents Canada. We particularly don't like to be on top because our health outcomes are really not that great, even though we spend more than every other developed country. But a growing body of evidence, as I hope I have tried to convince you today, has pointed to links between decreases in microbiome diversity and chronic diseases. And we also have links to suggest that our Western lifestyle, processed foods, antibiotic overexposure, C-sections, and a more sterile lifestyle than perhaps earlier generations led. These links to chronic diseases are there, and they're irrefutable. I'm going to skip over this. And so I'm going to then just leave with you the idea that it may be possible to intervene in ways to try and reverse some of these losses in microbial diversity. We know that various lifestyle factors have wreaked havoc on our microbiome, but we're also starting to investigate various interventions that may, in fact, reverse some of this. Things like probiotics and prebiotics, novel probiotics that aren't based on dairy products, fecal microbiota transplantation, and synthetic stool substitutes. And I will just go through all of this. Let me go back one. And let me just comment here that I think, like everything else that we've talked about, as we understand with how we react to prescription drugs, it is really unlikely that we are going to find a single intervention that is going to work for every individual in every circumstance. And I think it's naive to think that it would, particularly when we're considering such complex ecosystems. Fecal transplants have turned out to be extraordinarily useful and may quickly become the standard of care for treating antibiotic-associated diarrhea associated with C. difficile. This is a situation where repeated exposure to antibiotics has eliminated much of the resident microbiota, allowing C. diff, which is typically present in all of us, to overgrow. People die from C. diff, antibiotic-associated diarrhea, and up until fairly recently, the only treatment for C. diff, which is a bacterial pathogen, was even more powerful antibiotics. But that's what got the patient into trouble in the first place. But there have been some spectacular successes using fecal transplants, and a number of companies now are looking to see if they can take fecal transplants from where they are currently to something that's a little bit more standardized and a little bit more controlled, to the idea being that you could capture the bacteria that are essential for reconstituting the antibiotic-depleted gut and deliver these to patients in need in the form of a capsule. So this is an incredibly interesting area of expertise. And I think in the interest of time, I have more to say, but I think in the interest of time, I really need to stop there, so we do have time for questions. And let me just conclude here. And where are we today in terms of our understanding? Has the microbiome been overhyped, or is it a great hope? And I had some interesting conversations with the students before the talk about this. And I guess I would say it is some of both. So I would caution all of you, as you continue to read about the microbiome, particularly in the general press, to be skeptical of what you read, to think of what we've talked about today, that so many of the studies that have been completed so far really represent associations and do not yet allow us to address cause and effect. But this is an incredibly hot area, particularly in terms of microbiome therapeutics. The estimate is that in the United States alone, close to $400 million has been invested in setting up microbiome therapeutic companies. So obviously, the investors feel that with regard to the microbiome, it is a great hope. And I think ultimately what I am hopeful of is that a better understanding of the microbiome will ultimately allow us to identify certain patterns, certain species, certain metabolites that can be used as biomarkers of disease and can hopefully point us to entirely new therapeutics. But we need to be patient because the scientific process can take time. There are opportunities for you, if you're interested, to have your own microbiome sampled. This is just one of them, the American Gut Sampling Project. It's really quite simple. You're not going to get any information that you can act on, but you will see how you relate to an ever-growing number of citizen scientists who are interested in having this information. And I guess maybe I will just conclude with this slide, which is from this very peculiar comic strip called Toothpaste for Dinner. It's one of my favorites, and it really looks at the world through a somewhat skewed lens. But for those of you who can't read this, it says, "No, you can't have a dog. You have over 500 species of bacteria in your body. Why don't you go play with them?" So just be thoughtful. You are the keeper of an extraordinarily valuable ecosystem, and everything that you do affects not only you, but all of your microbial partners. So I hope that in the decisions that you make, you will choose wisely. And thank you very much for your attention today.
So, as was actually you were promised a slide how to hook up to Slido. We also have 2 mics on each side of the room. So please bring on your questions, and I have one for Claire. Actually, I had a bunch of them, and you answered them as you went.
Good.
So, but this one, so von Leeuwenhoek, 200 years later, you have Pasteur. About 100 years later, you have Dubose. About 50 years later, you have genomic sequencing. So what's the next breakthrough in 25 years?
Interesting question. It is my greatest hope and also my expectation that with a much richer understanding of the microbiome, this will become part of mainstream healthcare. I have to think that's true. Otherwise, it would be hard to keep doing what I'm doing. But I really think that as we have come to appreciate what an important role the microbiome plays in health and how it can, in some ways, influence outcome of disease, I have to say that we are not yet fully including all of the relevant information that we need in the practice of medicine. So there's an awful lot that needs to be done. We need to develop standards and reference ranges for all of this. We have to figure out what the most relevant measures are for the microbiome. As I said, I don't think it's going to be who's there. It may be metabolite profiles that we measure, much like we measure cholesterol today. But what I didn't mention is that we know that the microbiome, the gut microbiome, can play a role in metabolizing some of the drugs that we take. So imagine, and chemotherapy drugs are an example. Some chemotherapy, the same chemotherapy drug can be differentially effective in different individuals. It's not out of the realm of possibility that part of that difference might be because of what the microbiome is doing to metabolize the drug. So I think that as we gather more information and we can fold that into medical practice, we'll be much further ahead, and that's really my hope.
So really interdisciplinary approach.
Yes, absolutely.
Please.
Claire, thank you for the talk. My name's Dave Patrick. I lead our BC Center for Disease Control, and I appreciate what you say about correlation and causation. But over about 15 years, we've seen a 60% decline in the rate of antibiotics used in the population of infants in BC under 1 year of age. So would it surprise you to hear that the asthma incidence rate has fallen by 30% in that same cohort of kids?
Yeah, no, thank you for sharing that. I had heard that, I think, from Brett Finley earlier today. That's a spectacular success. No, it doesn't surprise me at all, and I think that really speaks to what I mentioned. And it's just those kinds of studies that we need to try and get at whether or not these correlations are real. So, I mean, congratulations. That really should be a model worldwide. So you are to be applauded, and those outcomes are really very exciting.
So on a lighter note,
Yes.
if you're a microbe for a day,
Oh, if I was a microbe for a day.
which one would you be and why?
Well, I guess it would depend upon my mood on a given day. Do I want to be a good microbe or a bad microbe? I think what I would probably want to be is one of these key butyrate-producing microbes, and I'll pick one, Faecalibacterium prausnitzii. I mean, I have become such a fan of butyrate, and it is so critically important to health. And you know, I went into science to try and do good. So I guess if I was going to choose to be a microbe for a day, I would choose to be one of the ones that really is known to have a very positive impact. Of course, if you catch me on a bad day, I might say I want to be C. diff, but not today.
Please.
Thanks for your talk, fascinating talk. I would like to keep things practical for myself. I have two kids, a 4-year-old and a 1-year-old. And the 4-year-old, she's very polite and she follows the rules, obeys the rules, and she always washes her hands before meals and all that. My 1-year-old understands no words. She eats my shoes when I'm not washing, like, everything. So I was wondering, where should we draw a line between, allowing toddlers do whatever they do because they need that microbiome in their guts, and when we grow up and become, older, do we really need to wash our hands if we just went outside and, sat in the car seat, or do we use a public washroom in the kindergarten? Things like that.
Great question, and I think the answer is I would err more on the side of your 1-year-old, what your 1-year-old is doing in terms of exposure to lots of different environments and lots of different microbes. I'm not in any way going to say that hand washing is a bad thing, but if you've come in from outside and you've just been playing in the dirt, it's probably okay if your hands don't get washed. That's a very different situation than if you find yourself with a bunch of people who are sick. That's not necessarily good exposure. So there, a little bit of hygiene is probably a good thing. So what I think where we've gone a little bit overboard, and I don't know if it's so true in Canada, it's certainly true in the US, there isn't any place that you go where there isn't a hand sanitizer available. And I see people using these all the time. And it's been shown that those can have an effect on the composition of the skin microbiome. So my thought would be, like so many things in life, you just want to apply a little bit of common sense. You certainly don't want to go looking to get infected with various communicable diseases, particularly this time of year, colds and flu. So there's probably no such thing as too much hand washing. But if you're in your own environment and you know that there are no dangers like that, I would say, you can probably be a little bit more relaxed.
So just to make it concrete, if she was playing in the house for about an hour, and then she takes a piece of bread, there's no necessity to wash her hands to do that.
You're putting me on the spot here.
We'll have to book a personal consultation for you.
Yeah, we will. But a healthy, fully functioning immune system is extraordinarily powerful. It's amazing what you can get away with.
So allow us to alternate a little bit between the microphone and the Slido. So keto diet.
Yeah, keto diet. I would not, based on what we know about how various macronutrients are processed, I would not recommend the keto diet over something like the Mediterranean diet. But there's a caveat. That said, what we do really now understand in a way that we didn't not that many years ago is that we are all different. So I am not going to make a blanket statement that there is no individual or no situation where a keto diet is not appropriate. I actually have a friend who runs a research institute in Pensacola, Florida. He suffered from severe epilepsy as a child, And there were reports that basically a keto diet could help keep epilepsy under control and eliminate the need for medication. His parents put him on a keto diet. He is now 60 years old. He's been following a keto diet his whole life, and he hasn't suffered from epilepsy. That's an extreme situation where there are other factors. But I think, I guess what I would say is we probably want to wait a few more years, because I'm really intrigued by what some of these companies are doing, looking at how the microbiome processes various macronutrients, how we as the host do. And I really do think we're going to move towards a situation where as much as it's sort of snake oil today, that there may be some reality to those kinds of personalized diets in the future.
Yeah, given the complexity of that ecosystem, it's really hard to kind of, if you don't really understand the pathways,
Right.
how can we understand the diet that connects to them, right?
Yes, exactly.
Please.
It's sort of a two-part question. Sure. One flows from that, and also what the gentleman was saying. My mother-in-law always said, you worry about the first one, and the second one eats out of the dog's dish. And I'm married to a very healthy third one. So one question's about, that follows from what you were saying about keto diet, is the Inuit diet, a traditional Inuit diet. And then the other one is tangential. Dr. Vikram Misra at U of S, who's a virologist, I know you use a lot of animal models. He looks at bats, and he says they don't have an inflammatory process. And they've got bats the size of hamsters that have been banded. They find them and they're 30 years old and healthy. They don't get any age-related diseases. Has there been anything you know of that looks at their microbiome and how it would relate to us and what we could learn from it?
Not that I'm aware of, but the literature is so vast. That's not to say that the studies haven't been done. I'm going to go do a literature search when we're finished today to see, because there may be something, really interesting there that we just haven't focused on. And, it wouldn't surprise me. It used to be in the very early days of the Microbiome Project, you could keep up with the literature. Now you do a search, Human Microbiome, and you come up with something like 30 or 40,000 papers that have been published. They're not all of high quality, but everybody has jumped on the microbiome bandwagon. So, but that's really an interesting observation. Germ-free mice who lack microbiome also show very low levels of inflammation. So, there are some So the microbiome can contribute both in a positive way, but if we mistreat it, it can turn on us too.
And can you comment, the keto diet is akin to the traditional Inuit diet?
Yeah, it really is. And I think there we have a unique situation where these indigenous populations have evolved somewhat in isolation, if you will, for a very long period of time. And so it's very likely that their microbiomes have evolved with them and are best suited to those diets. And so I think, again, we have to be careful about blanket statements, but to take someone who has been eating a typical Western diet with sort of a more even distribution of all the macronutrients, you then switch to a keto diet. It's not that your microbiome can't adapt, it will, but it may maladapt just because it's, it hasn't evolved over the same period of time.
Thank you.
So why don't we go over to the other microphone?
Thank you. I have a question about alcohol consumption. What does the research say about that in the microbiome?
Perhaps you can repeat the question.
Yeah, no, the question is, what do microbiome studies say about alcohol consumption? I think that in moderation, I am not aware of any data to suggest that alcohol in moderation poses a problem. That said, there are exceptions to every rule. And some interesting reports sort of on the flip side of this, that there have been reports of individuals who will eat certain types of carbohydrates and end up intoxicated because their microbiomes will basically create ethanol as a byproduct of metabolism of these carbohydrates. So But that's not, I know that's not what you're asking, but it's just an example of the diversity in the metabolism that we can associate with the microbiome. But reasonable alcohol consumption, and of course I'm not going to define reasonable for you, has not been shown to be a problem. Excess alcohol consumption, yes. And I think reasonable and excess is in the eye of the drinker.
You have been very patient.
Sorry, I just want to add to that real quick. Do you have 2 or 3, like your favorite books or other resources about the microbiome? Say, out of all of those books that you listed.
I think Brett Finley's 2 books are outstanding, and probably the third one that I would recommend, and I'm not entirely sure of the title, but the authors are Justin and Erica Sonnenberg. It may be The Good Gut Diet. But most of those that I put up there are not ones that I would recommend.
Thank you.
You're welcome.
Please.
Hello. While you were speaking, I had this sort of vision of a future when you could really take a tablet with the correct microbiome for a place you're traveling to. For example, if you wanted to be able to drink the water on one of those cruise ships. And even before that, of course, just, well, for example, I don't know, anything to do with skin sensitivity might be found to be inflammation related to lacking certain microbiota. So what I'm wondering is, what do you see as the biggest barriers to being able to go to the doctor and ask for a prescription for the lotion of the right bugs?
Right now, the biggest impediment is just understanding where those links are reality and where they're just coincidence.
I guess I'm asking for a pathway. What would you see happening between now and then that is what's going to make that possible?
Well, I think there have been a number of microbiome-related companies that have been started around the world. And some of these are taking some really interesting approaches. They're trying to mine the human microbiome for novel probiotic organisms. Faecalibacterium was the one that I mentioned that I would like to be for a day. There's a company in Boston looking to try and engineer Faecalibacterium to be used in various inflammatory bowel disease situations where you can restore some of the butyrate concentrations, as an example. And so I think it's those kinds of approaches. There are other companies that are mining the microbiome, not for organisms, but for the metabolites, the byproducts of these organisms, because these organisms impact human health. They have to do it through a particular pathway, and it may very well be that in many instances those are chemical signals that are delivered to us from our microbiome. And it's really quite interesting. In a previous scientific life, I used to work on G-protein coupled receptors that are cell surface proteins that are involved in cell-to-cell communication. This is a large gene family. And at the time that I left the field to start doing microbial genomics work, there were a number of orphan receptors that had been identified. We knew that they were receptors. We could find them on the surface of cells. We just didn't know what bound to these receptors. Well, it turns out an awful lot of these are in the GI tract, and they're not responding to signaling molecules that are encoded in the human genome. They're responding to bacterial metabolites. So I think that what we need to do is to really figure out what these signaling pathways are, whether we can mimic them through the use of specific organisms or through various metabolites. My guess it will be both. And once we get to that next level of understanding, then I think we can go forward to begin to bring the kinds of ideas and products that you described to market.
Thank you.
You're welcome.
So, last question.
Yes.
Actually, we'll combine two. We'll combine the question on probiotics and their use with the way to mitigate the effects of antibiotics if we have to take them.
Okay. Yes, probiotics. I'm actually doing a lot of my research currently is on probiotics, and I would say that despite a lot of studies that have been done, there isn't a whole lot of rigorous scientific data to give us insight about how probiotics work. A lot of what I and my colleagues are trying to do is to devise studies to better understand those molecular mechanisms. What I will tell you from the work that we and others have done so far is that a given probiotic does not behave similarly in all individuals. And I think based on what we've talked about today, that shouldn't be a surprise at all. And certainly the second part of this question, are there ways to mitigate the effect of antibiotics on the human microbiome when they are medically indicated? And this is a situation where there has been some evidence. Again, I wouldn't say it's the most rigorous evidence, but there has been some evidence that certain classes of probiotics can be of some benefit when taking antibiotics. We have some ideas, some hypotheses about what the mechanism might be. We're in the middle of a couple of studies trying to test those, and it gets back to maintaining the concentration of short-chain fatty acids and having probiotics feed some of these highly beneficial bacteria, even in the face of the onslaught of antibiotics. But it's still a little bit too early to say. I do think that if we can continue with this kind of research, we will end up in a place where we have a much better understanding of how different probiotics behave and how they behave in different ecosystems. So we can get away from this approach right now where probiotics are often prescribed or just consumed because we understand that these are generally safe, and even if they don't do any good in a healthy individual, they're probably not going to do any harm. And I would say that those are probably true statements, but for me as a scientist, that's extraordinarily unsatisfactory. And for those of you as consumers who are buying probiotics, it may also be unsatisfactory if you aren't sure that you're spending your money wisely. I've been encouraged by the interest that certainly in the US that the NIH has shown in trying to fund these studies. So again, my comment is we need to indulge you and ask you to be patient while the science plays out and we collect more data.
Claire, thank you very much. The abundance of questions shows that we not only have picked a subject that has really triggered a lot of interest, but we also chosen a speaker who has really presented it in such an engaging way that our brains were very much stimulated.
Thank you.
So thank you very much, Claire.
Thank you.
Don't go away yet. I'd like to ask the chair of our board of directors, Dr. John Thompson, to present you with the traditional Don Rix Keynote gift.
Wonderful.
I get to do the fun part. So I'm just going to make a few quick comments first and then make the presentation to you. So my name is John Thompson. I'm chair of Genome BC. And I have to say I feel pretty humble in that role, given that I am, in a sense, representing Don Rix here tonight. So that was an absolutely amazing talk. Claire, thank you so much for that talk. My background is actually earth science, so there's a certain learning curve that I'm on when I listen to talks like this. But I have to say that I operate at a very different scale on the earth, but the complexity is perhaps even nothing compared to some of the complexity that we've heard about this evening. It's interesting, Claire mentioned that bacteria and microorganisms have been around on the planet for several billions of years, and it's actually increasingly clear that they played some really important roles in Earth and early Earth history. So we have a bit of a common interest there. However, the glimpse you've given us tonight into our world, into our ecosystem, and the role that microorganisms, the microbiome plays in our health and our lives is amazing. It's a little scary, I have to say, trying to come to grips with both the complexity and the role of the individual. I've always thought individuals were the best thing that we could possibly be, but I wish we weren't quite so individual. Perhaps life would be a little simpler if we weren't so distinct in our own individuality. I'm fortunately, and Pascal would appreciate this, very early in my life I met yogurt in Switzerland, and I've been a passionate yogurt eater ever since. And so I thought I was really good about this. I thought I was way ahead of the game and clapping myself on the back for consuming so many probiotics, until you said something about non-dairy probiotics, and I went, oh damn, yogurt may not be the answer after all. Anyway, that's my simple summary of what was an absolutely amazing talk, and I'd just like to ask you again to thank Claire so much for that. So we now have a presentation, and this is the fun part. So Claire, as a gesture of our appreciation for sharing your insights today, I want to present this talking stick to you, which was carved by Coast Salish artist Jim Yelton. So the talking stick, as many of you probably know, is in indigenous cultures, is a sort of important and critical powerful communication tool. The person holding the stick, that's me right now, is the only person who's designated as having the right to speak, and all of you must listen. So this is a very valuable and important tradition and a wonderful piece of symbolism. It's ripe with spirituality and tradition, and each First Nation has its own culture and traditional history that's around it, so each one is unique, but they share some common attributes and symbolism in the carvings. So this particular talking stick includes the eagle that symbolizes leadership, great strength, focus, and prestige, and the eagle also represents friendship and peace to all. That's a wonderful idea in the modern world. The bear represents strength, family, vitality, courage, and health. I emphasize the health, given the topic of the talk this evening. And the orca or killer whale symbolizes harmony, longevity, and strong community. The orca is also said to protect those who travel away from home and lead them back to where they come from when the time comes. So that's very appropriate for Claire's travels tomorrow. She hopefully will be in good hands. So without further ado, I'd like to present you with the talking stick.
Thank you. This is absolutely magnificent. I'm teary-eyed. Thank you so much. I'm not sure I'm ever going to put this down. And I don't know whether I will use this more at home or at work. But thank you so very much. Thank you again to everyone for your fabulous hospitality, for your generosity, for your interest. This has been a phenomenal trip. And if I hadn't promised my husband that I will step away from my responsibilities 100% of the time in the next couple of years, I would have already put in my application for the open position at Genome BC. But, and I mean that in all sincerity, I actually had a conversation with him on the phone last night, and he said, if you want to move to Vancouver, we can do that. But I don't think that would be a wise move. But I really do hope that we can all stay in touch and that our paths will cross again. And I just want to applaud Genome BC for the enormous impact that you have had. I am truly envious. I don't know of an organization within the United States that has had such a far-reaching impact and has done as much good. And so you should really all be very proud of what you've accomplished. And thank you very much.
And thank you again, and we'd certainly welcome you back. And if any additional persuasion is required, I'm happy to make a phone call. Before we wrap up the formal program, I just have a couple of things I want to announce. I ask you to complete the short survey that you'll receive in the email, because the feedback that we get is vital to putting these events on and so on. And I'd like to acknowledge a few people. Pascal for looking after the Q&A, which was extremely well done. Sally Greenwood for the introduction at the beginning and for orchestrating this amazing event. And I guess the roundtables that happened earlier today as well. And of course your team, Jenny, Brad, Anna, Kim, all of whom played critical roles. And I'd like to also acknowledge Catalina, who worked with Sally to help organize this program before she decided to step away from Genome BC, with a great loss to all of us. And so in conclusion, the keynote portion of our program, we'd like to encourage you to stay with us and join us for discussion outside with some refreshments. And thank you all for coming again, and thank you all for attending what has been a super evening and event. Thank you.